Thanks so much, Aubrey. That's a pretty good summary of how I got where I am today. And Aubrey can claim some no small amount of credit for that. So I first learned about the problem of oxidized cholesterol, which is most of what I'm going to talk to you about today, from Aubrey, even before the events that he's just talking about. Back when I was a graduate student, he came to speak at my university and actually gave a couple of lectures on a couple of different topics in some different universities and departments around the area, and I followed him around a little bit. One of the things that he taught me about was this idea of oxidized cholesterol, that when a free radical, an oxygen-free radical, bumps into regular cholesterol, it more often than not turns into 7-ketocholesterol, which is very toxic and involved in multiple aspects of aging. I'm going to talk almost exclusively today about the cardiovascular disease, atherosclerosis, which is what we've been focusing on in trying to bring our first drug to clinic. But what I want to emphasize is that this is a basic mechanism of aging, one of the basic kinds of damage that accumulates with age in various cells and tissues. And so the mechanism, the model that we have for atherosclerosis, which is the thickening, the clogging of the arteries, is that macrophages, their job is to eat, and in atherosclerosis, they go to the site of a lesion, and their job is to eat up the junk that they find there. And they're very good at their job. But when they eat up too much of the lipids and other junk that they find, they start eating up a little bit of 7-ketocholesterol, 7-KC, and just a little bit of that, once you get enough of it over time, is enough to shut down the lysosomes of the macrophage, which is the garbage processing unit of the cell, and the macrophage then can't metabolize really much of anything anymore, and not the lipids, and they balloon up. They keep eating, and they balloon up into something called the foam cell. And the foam cell layer of the plaque is a fundamental step in the progression of plaque. And our goal was to see if we could get rid of the 7-KC and thereby rejuvenate the macrophage and see if we could put it back to work, doing what it's supposed to do. I'll just take a little bit of an interlude to talk about how people have been treating cardiovascular disease and developing therapies. There's drugs for trying to reduce the likelihood of having a cardiovascular event, like blood thinners. Then the next generation of drugs is lipid-lowering therapies, which now we're going on about our third decade of those. Then the next generation of lipid-lowering therapies, which are better at lowering lipids, but not really doing anything fundamentally different. Now we're actually starting right now. What's starting to come out is the anti-inflammatories, which I think are starting to address some of the actual damage problems that are going on, and some of the genetic problems with things like Lp(a). But now I want to push us into real damage reversal therapies, and that's why we're targeting 7-KC in cardiovascular disease. And just a little bit, you're starting to see some good quality clinical data come out about association of 7-KC with cardiovascular disease. Not the same level, so there's maybe 3 good, decent-sized studies of 7-KC and cardiovascular disease, as opposed to, say, like 1,000 with LDL. But I think the association, not just with correlating with disease, but also risk association that the more 7-KC you have, the worse off you are, makes me think that we're in a good position and at a good time in history to finally, almost maybe 20 years after I learned about 7-KC, to actually be doing something about it. So back at the foundation, like Aubrey was mentioning at the beginning, we were working on a lot of fancy enzyme engineering approaches that were cool, but I started thinking, trying to think of ways that I could drive something to clinic really fast. And so I dug into the literature and found these molecules called cyclodextrins. Cyclodextrins are cyclic carbohydrates, and there was a lot of work back in the 80s and 90s that were looking at the cholesterol and oxidized cholesterol binding properties of cyclodextrins. So we started playing around with them there at the foundation, got some assays working, and then started modeling them computationally. And I didn't bring any of the computational data with me today, but the earliest modeling that we did showed that the best binding of 7-KC happened when you took two medium-sized cyclodextrins, beta cyclodextrins, together in a head-to-head configuration, and that was the most stable complex. And so once we started getting things working, we asked ourselves, well, if that's sort of the natural way that these things come together, what if instead of taking decades-old cyclodextrins off the shelf and playing around with them at very weak potency the way most people do with cyclodextrins, what if we built new ones? And what if we stuck them together and dimerized them the way and just forced them to emphasize that conformation? And long story short, we did that, and it worked exceedingly well. And we were able to invent new cyclodextrins that were about 1,000 times more potent than the stuff you could get out of the chemical catalog, and we could engineer them for high specificity for the for 7-KC over regular cholesterol, and that turns out to be really important for making them efficacious, but also safe. We can make a lot of cyclodextrins. We have a lot of prototypes on the shelf that are very potent, but bind cholesterol really well, and they're all really toxic. If you strip too much cholesterol out of a cell, that cell falls apart instantly, and you'll get toxicity from that. So we carefully engineered our cyclodextrin to have a strong preference for 7-KC, and then we did what we set out to do, which was try them in foam cells. So we took some macrophages and grew them on a plate, and then gave them just a little bit of 7-KC that you can see in the bottom group of cells there, and you see that red sort of blotchy droplets in the middle. They're called lipid droplets. That happens really quickly when you give them just a little bit of 7-KC, and then when we give them our drug, they quickly go back to looking normal. And they don't just look normal; they start behaving better again. So we can take macrophages, we can take mouse macrophages, we can take human macrophages, we can treat them with 7-KC, we can treat them with ox-LDL, and we see the same kind of phenomenon that I'm showing here, just a few examples of. They not only look better, but they start acting the way they should act. The job of a macrophage is to eat. They eat less when they are poisoned with 7-KC, and they turn into foam cells, and their activity comes back when you reverse that with our lead drug, which we call UDP-003. I'm not going to go into the massive amount of profiling that we've done on these cells too much, but I just put in a couple of examples of some gene expression profiling that we've done to look at some key pathways in lipid metabolism that get shut off when the cells become foam cells and get turned back on when we rescue with our drug. And then another thing that happens is that macrophages turn on a signal that says "don't eat me," the same way some cancers do to resist the immune system. Another company is trying to target this particular cell surface receptor to target atherosclerosis, and so they're targeting this CD47 gene in particular. And so we wanted to know whether we were also affecting that, and we are indeed able to at least bring that down to close to normal levels again. And one more thing, some brand new data that I think you guys are going to hear about first here is that we've been profiling the inflammatory cytokine profile that macrophages give off when they are normal or when they're foam cells, and then when we rescue them. And this is just two of the most dramatic examples that we've found so far. Is that IL-8 and MIP-1 alpha pro-inflammatory cytokines get super hyperactivated. So this is actually fold activation, not percent increase. And so these are the two most dramatic examples we found, is that IL-8 is getting like over 25 times upregulated when they turn into foam cells, and then we're rescuing that by a pretty remarkable amount with a very short-term treatment with our drug. So that's the main bit of the science that I wanted to focus on today. And what's most exciting is that we're not just an R&D company anymore. We're transitioning into being a clinical stage company, and we're getting ready to enter clinical trials hopefully later this summer. So this is our clinical trial plan. Or overall clinical trial plan. I'll go into a little bit more detail about these different, at least the first two steps in the next couple of slides. We're going to do our phase 1 in Australia, in Adelaide. And it's going to be primarily a safety trial, and we'll be able to look at some biomarkers and some patients. And that will lead directly into a phase 2 trial where we'll look at a much larger number of patients, looking directly at efficacy on coronary artery disease of the plaque. And so non-invasive imaging of atherosclerotic plaque has really advanced in leaps and bounds just in the last 5 to 10 years. So we're going to have some fantastic opportunity to look and see whether we can actually regress plaque in humans in just the next few years. And then if we can, there's lots of indications that are open to us to go into for approval. So we can go for a smaller indication like peripheral artery disease, or we can look at chest pain, angina, or we can go for the biggest type of indication that we can imagine, which would be like full coronary artery disease with looking at reducing numbers of heart attacks and strokes. So that's our strategy. We have, importantly, to get here, we had to make an actual drug product. And that's way beyond, Utah talked a little bit about some of the challenges and how important it is, but I think he underplayed a little bit how challenging and time-consuming and expensive it is to take a drug, whether it's a cell or gene therapy product, or a small molecule drug or kind of a medium molecule like our drug, and manufacture it at high quality at high scale that is safe for people. So we managed to scale our manufacturing to the kilogram scale and create a liquid formulation, and now it's ready in single-use vials, sterile individual-use vials for patients. We have our first cGMP batch done. We've had many interactions with regulators. We've met with regulators in the UK. We've had our pre-IND meeting in the US. And this week, we're submitting in the next few days, end of this week or beginning of next week, our clinical trial application in Australia, just putting the final edits on the protocol. And we've finished all of the GLP safety studies. So all of the small and large animal testing is done. Our drug is safe. And it's ready for people in phase 1. So we've got some phenomenal. We've really reached into the, like Aubrey said, I come really strongly from the aging field. I only worked on aging when I was an academic, and I went into the foundation, and of course only worked on aging when I was at an Aging Research Foundation, and now we're spinning out a company, and we're focusing on cardiovascular disease. So that's been a learning curve. And part of that has been bringing on some top cardiologists to advise us. And these are some of the biggest names in cardiology. And most importantly, that first one, Steve Nichols, is the biggest cardiologist in Australia, and bringing him on board really opened up doors for us in Australia, helped us get introduced to people that we needed to know to make the clinical trial happen quickly, to get to work with the best people. And he's co-directing our phase 1, which for the kind of guy who usually gets called to oversee the phase 3s by the giant pharma companies, we're really lucky to have somebody like him helping run our phase 1. So a little bit of detail about our phase 1. It's going to have 72 healthy volunteers. And as soon as that's done, we'll get to work on actual patients. And so we'll be recruiting 12 patients with coronary artery disease to participate in what you could call a phase 1B portion of our phase 1 clinical trial. That's all planned out, and like I said, hopefully starting within 2 months here, maybe 6 weeks. The phase 2 is a bit more conceptual. Tentatively, we're looking at the same patient pool as in the 1B, the same indication group, but expanding to 150 patients at a placebo and a low dose and a high dose group, and tentatively flagshipping that at Cedars-Sinai in Los Angeles. And so that trial will begin, hopefully in something like a year and a half, and to be finishing up by around the end of 2027, beginning of 2028. And if that succeeds, that will be our let's call it our ChatGPT moment. If we're able to see patients with their arteries, their coronary arteries, half or 80% occluded, and have that drop way down, below 50% occluded, that will blow the lid off of this. That will be when the bidding wars start from the big pharma companies, when everybody sits up and takes notice and realizes that you can target a basic aspect of aging and reverse a fundamental disease of aging, and that's the moment that we're really looking forward to. So just a little bit before I finish up on other applications for our drug, we're doing some small R&D projects on stroke recovery and Alzheimer's disease and some orphan disease. And we're also interested in applying our technology, this cyclodextrin technology and the cyclodextrin dimer technology, to other disease targets. Things like bisretinoids that accumulate in the eye, causing macular degeneration, other oxysterols, and other targets as well. And our technology is also applicable to things that aren't in aging. So of course it's technology, and so we think it could be valuable for overdose reversal, for anti skin rash stuff, like things like poison ivy, things like that. We're really hoping to be partnering with experts in other fields, and we're starting to have experts in those fields call us and say, "Can we use your technology for our problems?" And we're really looking for partners and partnerships in those realms. And we're also looking for other ideas of things that we can target, ideally things that are relatively small and hydrophobic, so things that are, say 1,000 or fewer Daltons in size, and ideally hydrophobic, that we can target with our technology. We have this powerful computational platform that we've built, where we can take any target and throw it in and model cyclodextrins and make predictions about new ones that we can bind with them. So give us your ideas and we'll try them. And we have a new AI training algorithm that we're training using thousands of simulations that we've already performed, that we hope to be able to use, so that it doesn't have to be humans anymore coming up with the ideas for the structures of the drugs. We just plug in the target and ask the algorithm to tell us what the best drug to make, and then we can make a prototype and try it out. So I'll wrap up, I think, with plenty of time for questions if the audience has any. Like I said, if you think that my drug can work in your model system, let's collaborate. We can do an MTA and I can send you samples. If you're interested in investing, come and talk to me. We do have a funding round open, and we need a little bit more money to fund these clinical trials. So please say hi. And this wouldn't have gotten anywhere if it wasn't for the wonderful group of 12 people that you see on the slide there, and a small army of consultants and CROs that help make this happen. So thanks, everybody.
Yes, and as Aki says, there are a couple of minutes for questions. So, yeah, I didn't bother with questions during the orientation session, but we will have them throughout the rest of the meeting. We have, as you can see, a couple of volunteers who have roving mics, so just make yourself visible to them, and we will be nicely efficient. Yeah, so I'm just going to ask the first question while I'm here, and then we'll have... So there's been some reports over the years of toxicity of the cyclodextrins for hearing. Have you done any tests of that with your dimers?
Yes. Thanks, Aubrey. This was something that we were really sensitive about, and it turns out that cholesterol problem is the main issue that you need to worry about. And so when we tuned our drug to be selective for 7-KC, that took care of the ototoxicity problem. And there's some fantastic in vivo tools for really extremely sensitively measuring ototoxicity, which is hearing loss, in rodents. And we're going to have a really, really wide therapeutic window. We can overdose rodents and damage hearing, but if you stay below the threshold, you don't damage hearing at all, no matter how many times you dose.
All right.
Thank you. 7-KC also can come from food. So is your solution help to get rid of those 7-KC?
Maybe. We actually want to start a new project to test that idea now. In general, eating 7-KC is not dangerous. If you have a young, healthy liver, and you eat 7-KC, then you metabolize it really well. If you don't, then you might have a problem with it. And there is some evidence for that in rodent models of liver disease. And so we want to look at that and see whether we can reproduce that problem with 7-KC and liver disease and then rescue it with our drug.
Yeah, so I was really excited in past conferences to see the awesome mouse data, the awesome computational data, and the platform being developed. And 7-KC is an amazing target, but it sounds like it's kind of particularly human, and it also sounds like it'll take a lot of time for you to get to the market because of all of the regulatory challenges and all of the forms, all of the processes you have to go through. And there are companies, for example, in the veterinary space that sort of cut this short, such as Rejuvenate Bio and so on. I was wondering, have you ever discussed with people in the veterinary longevity space about sort of like a 7-KC for cats or dogs or anything that you can get faster than a 5-10 year timeline to the market?
We've thought about it. We haven't had serious conversations about it. If there's anyone in the audience from from any of those organizations, I'd love to talk to you. There isn't much data on cats and dogs being essentially carnivores that are really good at metabolizing many cholesterol species. So it's not clear that they actually have a problem with 7-KC. They might, but there's not data in the literature for it. It's certainly something we'd be interested in exploring if people have something we can collaborate on.
All right, thanks very much.